Hello students, welcome to SK Biology. In today's video, I am going to discuss deviations from mendelism, also called as non-Mendelian inheritance. Incomplete dominance is one of the important non-Mendelian inheritance, which is well studied in genetics. Today, we are going to learn the concept of incomplete dominance in detail. Let us start. We already know Mendelian inheritance, in which we studied that alleles for any trait are present in two forms. One is dominant with pair of dominant allele and one is recessive with pair of recessive allele. In heterozygous condition, one allele will be dominant and one allele will be recessive. The presence of one dominant allele suppresses expression of recessive allele in case of heterozygous condition, and this phenomenon is called as complete dominance. Now the question is there any deviation or exception to this Mendelian inheritance? The answer is yes. There are few inheritance patterns in nature which do not follow Mendelian inheritance principle. We call them as a non-Mendelian inheritance. Today, we are going to discuss one of the well-studied non-Mendelian inheritance known as incomplete dominance. Incomplete dominance is a pattern of inheritance in which neither allele is completely dominant over the other allele, resulting in heterozygous phenotype being an intermediate. In this phenomenon, trait or allele shows deviation from Mendelian complete dominance concept. Let us understand incomplete dominance with popular example of 4 o'clock plant, whose scientific name is Mirabilis jalapa. 4 o'clock plant has different colored flowers. If parent plant with red flower is hybridized with another parent having white flower, it produces F1 generation plant with pink flower. This suggests that here no any gene or allele for the red or white color is dominant over other color. Both alleles are expressed equally and produced intermediate phenomenon, that is pink flower. So incomplete dominance is the result of mixing of two traits forming intermediate phenomenon. This trait is different than that of Mendelian inheritance pattern. Now suppose parent plant, which is red, is a male and alleles needed for red color phenotype is pair of R1 allele. In a same way, other parent with white flower is female and white phenotype is due to presence of pair of R2 allele. Hybridization of these plants will produce gametes R1 and R2 respectively. These gametes will fertilize to produce F1 plants with pink phenotype, in which both alleles are present, forming heterozygous condition. Expression of R1 and R2 allele is equal. No suppression of any allele as that of Mendelian inheritance. To study incomplete dominance for F2 generation, pink flowered F1 hybrid will be self-fertilized. First, it will give pure gametes R1 and R2, and after fertilization, different combinations will be formed, which we can see here in Punnett square. In F2 generation, total 4 plants will be produced. 2 plants with pink phenotype showing incomplete dominance. 1 plant is same as that of red parent and 1 is same as that of white parent. So, phenotypic ratio is 1:2:1. This ratio is different than that of Mendel's standard phenotypic ratio for monohybrid cross, which is 3:1. This deviation is because of non-Mendelian inheritance. Genotypic ratio for incomplete dominance is same as that of phenotypic ratio, that is 1:2:1. As that of plant, few animals also show incomplete dominance. Here I have taken examples of chicken and horse, where black colored chicken when crossed with white colored chicken, it gives rise to grey colored chicken. And in case of horse, brown colored horse when crossed with white colored horse, it gives rise to light brown colored horse. Both example shows incomplete dominance. I hope you all understood very well the concept of incomplete dominance.